Thank you, David. I want to start by congratulating Ralph on getting this award. Can't think of somebody more deserving than yourself. For those of you who don't know, I guess as David alluded to, Ralph, I was at Hopkins for 12 years before coming here. Ralph was my mentor, my advocate, my teacher, my friend. And I'm excited to, and I agree with Olja, coming up with something that Ralph doesn't know about pancreas cancer is very difficult. So this is what I came up with, and it's actually new for me. I just put the talk last night, I admit, and it's interesting. I think it's a little provocative, and Ralph, I hope you enjoy it. So. It's now 6 years ago. This is work that I completed at Hopkins showing that there is intratumoral heterogeneity of pancreatic cancer. Without going into what genes are heterogeneous, if you take different pieces of a primary pancreatic cancer and sequence them, the constellation of mutations that you find are different. We also found that metastatic subclones originate in the primary tumor and seed other sites. And this was published in companion with a paper. It was two papers. They were back-to-back in the same issue. So this was done in collaboration with the Sanger, the same samples. They used a different method. We had sequenced for SNPs. They had sequenced for rearrangements, but essentially the conclusions were the same. As a result of that work, over time, our lab, while it remains focused on pancreatic cancer, we've really become very heavily focused on cancer evolution and fundamental processes of cancer evolution. So, of course, there's the medical donation program that I did at Hopkins, and we continue here. One of the things with the program is, in addition to pancreatic cancer and understanding the patterns of evolution that we see in pancreatic cancer, we're looking at other tumor types in collaboration with the other DMTs at MSK to look at if those patterns are differ based on tissue type of origin or things like that. We have cross-disciplinary collaborations. So being interested in evolution, I thought, who knows evolution better than people at the Museum of Natural History? So I managed to freely weasel my way in. Even though I have a membership, it's still, I just love that I can get in without showing my card. And members of my lab and I, we've been meeting on a semi-regular basis with the chair of paleontology. And that's great fun. It takes a half hour for us to understand each other first, because they don't know anything about cancer and I don't know anything about paleontology. But once we understand that we're talking about the same things in the same terms, it's actually very fruitful. Right now, the exchange of information is all them to me. But I'm trying to convince them to come here for a visit to stay. So that's actually been very, very fruitful in giving insight into evolution, because we think of cancer evolution as just a form of evolution. So you can just borrow the same principles and tools. They actually have many bioinformatic tools that they've been giving us. We've been working on developing novel bioinformatic tools and pipelines. You're wondering why there's a jar of jam there. That's because the pipeline is called the jam pipeline in the lab, and that's named for Jungwei, Alvin, and Mark, who put it together. So we affectionately call it the jam pipeline, and it has its own little icon when you open it on the computer. But all of these methods are characterizations. It's looking at a snapshot in time of what's the evolution of a tumor. We're interested in getting into, are there any functional systems we can use to understand evolution? Start perturbing some of these features or microenvironmental factors to see what happens to the evolution. So one thing we've done is we have, through Eppendorf, acquired a system called the DOS-Box. It allows us to do long-term evolution experiments of mammalian cells. That's never been done before. We've had the system for about a year now, I believe, and it's up and running, and it controls every feature you can think of the canister and the cells growing in it, so you can change the glucose by 0.5%. And if you have your cells barcoded, you can see how different subclones respond to that. So you can look at how different genes affect subclonal development or stasis or regression. You can look at, you can do competition experiments, things like that. So that's a functional system. And then there's mouse models. The question was, can we use mouse models as a functional system to understand evolution? So for those of you who are not familiar with mouse models of pancreas cancer, and I certainly was when I started this project, there's many mouse models, but the classical model is known as the KPC model. And this model is a mutant KRAS allele and a mutant P53 in the mouse, both of which are activated in the same cell by Cre recombinase in a cell that is destined to become of epithelial lineage or ductal lineage. So this is referred to as the KPC model, and a lot of times it's the most common model used. It's thought to very well recapitulate the histology and the clinical features of human pancreatic cancer. The mice get hemorrhagic ascites, many metastases to liver. This is a pannin. Here's the infiltrating cancer with stroma, and here's even a metastasis. So it's a very commonly used model, well-regarded model. This model has been used to make many discoveries that have led to clinical trials in pancreas cancer. This is just representative, and I don't mean to leave anybody out or anybody's work, but for example, the discovery that the stroma may act as a barrier to chemotherapeutic delivery. The distinction between cells disseminating versus cells. Don't worry, Vulcan, I can give you the presentation. You don't have to take it. The distinction between cells disseminating versus actually colonizing and forming a metastasis, the enzymatic ablation of stroma as a way to deliver chemotherapy more effectively. The stroma as a tumor suppressive entity. There's been shown that if you remove the stroma by a variety of different methods, it actually restrains, you have a reduction in tumor growth. More recently, RUNX3 was identified as an important metastatic switch molecule. And finally, we just published, using this model, it was just published online last month, looking at mechanisms of hematogenous metastasis using this model. So it's been very useful for the field. I wanted to know, do they undergo subclonal evolution? Because I'm looking for a model system for our evolutionary studies. I actually went back to the original papers, and this is the original report of the KPC model, and they actually say in it, by several different methodologies, no mutations in the other principal tumor suppressor genes were apparent. Now, in fairness, this was 11 years ago. You know, it was a very different era in terms of sequencing. They looked at about 3 or 4 genes by Sanger and a number of other methods. And there were no mutations found in those genes, so that was fair enough. But that has led to the general thought that in this model, there's no additional mutations that occur beyond the transgenes that are used to create the cancer. So we proceeded to sequence these, and we looked at 2 groups. First, we used 6 KPC mice. So this is their genotype. KPC mice typically have a median survival of about 6 months. They have metastases to liver, lung, peritoneum, lymph nodes, just like a human being. The KPTC model that we created and reported in Clinical Cancer Research last month, it's the same model, but it's missing 1 copy of TGF-beta-R2. So just by making it haploinsufficient for that receptor, you actually get a robust change in phenotype. The survival is shorter, but the tumors actually are less metastatic. It's therefore a nice model of oligometastatic disease for pancreas cancer. This phenocopies if you do the exact same thing, but instead of the TGF-beta receptor, you take out a copy of SMAD4. And there is the reference. So this is what we did. We do multi-region sequence of human tumors. So we did mini multi-region sequencing of mice. So we took a mouse tumor, and the tumors are small. They're often about just 1 centimeter, 1.5 centimeters, and we cut it into 3 pieces. And we performed whole exome sequencing of pieces 1 and 3 to make sure they were spatially distinct from each other. The mutations that we found in those 2 were then used for targeted deep sequencing of all 3 pieces to validate, and then we also did array CGH for copy number. So that was the design of the study. This is essentially what the raw data looked like. So we achieved 133x mean target coverage over the mouse genome. We found 909 candidate somatic mutations based on the initial data that was produced. We used orthogonal validation, which is the targeted validation using a chip capture-based method so that you remove the artifacts that might have from the first form of sequencing, and that ended up with 159 validated somatic mutations. And those corresponded to 5 to 30 mutations per mouse tumor, or 1 to 28 mutations if you're looking at it by independent tumor region. This is the summary of the types of mutations identified. You see, as for human, most commonly a single nucleotide substitution. That was the most common form that you saw. And they were both silent and coding changes. That was most common. And you see there are some of the more classical deleterious type mutations seen. So this is similar to what you would see in human pancreas cancer. This is just an overview of the copy number data that was identified. For example, you see that there are examples where an entire chromosome was gained or lost. And you see in this case, it was present over all 3 pieces of the tumor, whereas there were some instances where there were focal copy number alterations, either gains or losses, and that was just present in this case in 1 piece of the tumor. So this is the summary of the somatic alterations per mouse, looking at both the single nucleotide variants and focal copy number alterations. And focal copy number alteration was defined as 5 megabases or smaller. And as you see, again, it recapitulates what we see in human, in that, generally speaking, most alterations within a mouse are mutations, and amplifications in some mice can be more frequent than others. So that's entirely like the human story. Next thing we did, though, with mutations, though, was annotate them, because mutations are good in one respect simply for evolutionary studies, but we wanted to know what are these mutations. So they were run through a number of complex algorithms. They're called SIFT and VEST, things like that, to see which ones are functionally significant. And about less than half, roughly 40–50% or so, were predicted to be deleterious. So this is how they would break down. These are the 12 core, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, well, we reduced it to 10. These are the core signaling pathways described by Jones et al. In Science in 2008, I believe. And you see, you know, with a mouse model, unlike humans where each human is different, and in that original paper, it was reported that each tumor had a mutation but the mutation that affected this core pathway differed, so each person had a mutation affecting all these different pathways. Because this is a mouse model, it's almost like a Groundhog Day experiment where you have those, almost like the same individual getting cancer over and over again. So you see, not every mouse has a mutation corresponding to every pathway, but collectively, there is a mutation in every pathway in at least one mouse. And these are the genes that correspond to. If it's in red, it's an amplification or deletion. Mostly we saw amplifications, and if it's in blue, it is a mutation. And you see it's cancer genes, FAT4, JUN, SMO, MYC, P53. So these are cancer genes that we can recognize. The ones that you don't necessarily recognize, it might be, in some instances, mouse genes are called different from the human ortholog. But there are things that unequivocally play a role in these processes. So then we want to look at evolution. So we classified these variants, and this is all variants again, all validated variants, not necessarily just the deleterious ones, by their evolutionary timing into ubiquitous, partially shared, and private. It's the same thing that we do when we do human studies. And again, you see there are most of the mice, all but one, had mutations that were ubiquitous to all. We did not include KRAS or the P53 or the TGF-beta-R2 transgenes, because every mouse potentially has those. So these ubiquitous does not count that. In one mouse, we found, based on the sequencing, that this was likely 2 pancreatic cancers that formed one sphere. They probably both developed and collided, and histologically it might have looked, it didn't really look histologically any different. And it's very common in these tumors that you can have regions of moderately differentiated and anaplastic and whatnot. So we figured this is actually 2 independent tumors in this mouse. In this mouse, there were only ubiquitous mutations, making us wonder if whatever happened caused a very rapid growth faster than additional mutations could accumulate, and the mouse died. Oh, this didn't translate, but that's okay. So this is, we then looked at how many subclones, the figure doesn't look the way it's supposed to, but it still works to get the point. We looked at how many subclones were present per tumor sample using all these mutations and the fact that different mutations might have the same allele frequency indicating they arose at the same period in time in the evolutionary history of the tumor. So for example, you see there's 1, 2, 3, 4 subclones in T1, there's 3 in T2, and there's 3 in T3. So actually you can get an idea of how the subclones are distributed across the mouse tumor. And we found, just like in human, there are subclones and they are geographically distinct from each other. So that helped. We then went one step further, because even if you can identify subclones using a method called PyClone, which we did, PyClone identifies how many subclones are in a sample, but it does not order them. So you don't know which subclone might have come before or after another. So we used a method called schism. And schism showed that the frequency by which KPC pancreatic cancers undergo linear versus branched evolution is roughly equal. So 5 of them underwent a linear evolution with an average of 3.6 clusters, mutational clusters per tumor. And you see in 4 of the 5 mice, these clusters are actually associated with the acquisition of a driver event. So again, agreeing, we're going back to the mutations that we have functionally annotated as being truly deleterious. We did not find anything in this one. There either was no driving event or we just did not detect it using the methods that we used. And these are example of the branched evolutionary trees. So there's more clusters when you have a branching evolution, but there's actually the frequency by which you see deleterious events is much lower, suggesting to us that branch is much more of a random type pattern, whereas linear is something that is progressing due to defined events. So what we conclude is that, yes, genetically engineered mouse models of pancreatic cancer do undergo subclonal evolution beyond the transgene events, and it can be used as a functional model, in my opinion. Examples, you can look at the relationship of different transgenes to clonal evolution. We did not find a difference between KPC and KPTC, but that's likely a numbers issue. There were some very interesting trends that I think if we add more mice, they might reveal themselves. You can use this to look at the relationship of treatment to subclonal composition within a single genotype. The question of the stroma constrains or promotes subclonal evolution. That is an interesting question. As I indicated, we didn't find, the reason we looked at the KPTC model instead of 12 KPC is because the TGF-beta pathway, no matter what part of the pathway you inactivate in a mouse model, it's a very robust, important signaling pathway in pancreatic cancer. So we cherry-picked the mice so that the KPC all had very aggressive metastasis, and the KPTC we studied all were oligometastatic. They either had none or 1 or 2 metastases. So they were very different clinically, mouse clinically. And even with that robust difference based on a single transgene, it did not translate into differences into mutational burden, mutational spectra, or the genes targeted during subclonal evolution. So we can at least make already a conclusion about that. Linear evolution was associated with a decreased survival in the mice. That was statistically significant. And depending on how you categorize the genes, there's a higher frequency, there's definitely a higher frequency of deleterious somatic alterations in cancer genes, depending on how conservative or lenient you are in categorizing them. The p-value wavers from 0.07 to 0.02. So you can imagine which way I'm going to lean when we write the paper. And different from, it's a very important caveat to point out, is treatment-naive pancreatic cancer. We have a paper that was just accepted last week. There is no accumulation of driver genes during progression and metastasis, whereas in this model, you do see that. So even if you use this model as a model of subclonal evolution, that's an important caveat to keep in mind. With that, I'll end. This is my lab. This work was done, it was a collaboration with Rachel Karchin at Hopkins and her very talented postdoc, Nushin Niknafs, who created all the algorithms and analyzed the mouse data. And many of the analyses had to be created for this project. And Yi Zhang, who came with me from Hopkins, is the other lead person who did this project. So he and Nushin are co-first authors on the study. And with that, I'll end. Thank you.